Good morning. I'm Michael Dong of MWD Consulting. My topic today is on the CMC processes of small molecule drugs and biotherapeutics. Here is my agenda. First, I will summarize the key differences of small molecule drugs versus biotherapeutics, and then describe what is CMC and why it is important. Second, I will explore the CMC processes of small molecule drugs from the API process chemistry, formulation to analytical development and QC, and then compare those to the biologics using monoclonal antibody or MAB as a model, MAP as a model. I will also compare the respective quality attribute and testing methods. More detail to be followed on the next webinar. Here's a high-level summary capturing the key differences of small molecule drugs versus biotherapeutics from the perspective of physicochemical property and pharmacological property. Small molecule drugs are typically a single high purity stable compound with molecular weight of 300 to 700 AMU, produced by organic synthetic route and purification by recrystallization. Drug products are typically tablets or capsules. Biotherapeutic using monoclonal antibody as a model, a complex heterogeneous biomolecule produced by recombinant DNA technology using fermentation and bioprocessing, purification, typically by chromatography. Molecular weight is 150 kilodalton. Drug products are parental only. Pharmacological property, for small molecule drug, the drug would binds to enzymes, receptor, or intracellular market, versus monoclonal antibody specifically binds to antigens of extracellular target. PK are very different. Small molecule in hours versus days or weeks for monoclonal antibody. Dosing is based on maximum tolerated dose for small molecule versus using efficacy biomarkers. And dosing frequency, daily is the norm, while weeks are the norm for monoclonals. The CMC, next we will discuss the CMC processes in drug development and why it is important. First, we will define what is CMC. CMC stands for Chemistry Manufacturing Control, which is a framework that ensures the drug quality of drug product is consistently produced and meet quality standard. Chemistry means the characterization of drug substance, structure, purity, stability, and physical chemical property. Manufacturing is the development of processes used in synthesis, formulation, and packaging of the drug. And control means quality control, which quality control staff would develop specification, testing method, and in-process monitoring to ensure the identity, strength, and safety. The CMC processes are important during drug development to ensure the clinical trial material CTMs are safe and efficacious. CMC activity supports regulatory filings and mitigate risks such as process variability and drug degradation. CMC often refers to an organization such as the Pharmaceutical Science Department or Technical Development Department in a pharmaceutical company. Here is the CMC organization, Pharm Science Department of a typical company, using the three pillars of process chemistry, formulation, and analytical development. Shown also here are some peripheral associated functionality and departments, such as PKDM, pharmacology, safety, toxicology, regulatory affair, QA, QC, project management, manufacturing collaboration, and clinical supply. Those labeled in red are roles played by analytical chemists. Note that each drug development project is typically driven by a project team and also by a CMC sub-team formed a few months before a drug candidate is nominated. Analytical chemistry and QC are pivotal in the CMC processes in new drug development, and here are their function and responsibility. Analytical development, characterization of the physical chemical property, perform method development, validation, and transfer for the various methods, provide analytical support for the API process and formulation development, initiate stability evaluation of drug substance or drug products, and collaborate with the CMC project team with also external CRO or CMO to resolve issues. The quadro control function is to conduct release testing and provide CFA for raw material, starting material, drug substance, and drug product, and to conduct stability study to establish shelf life, packaging, and storage conditions. Finally, to develop specification for QC of critical quality attributes and to contribute to CMC sections of regulatory filings. This table summarizes the characterization and QC tests for drug substance and drug product. Note that many of the tests are HPLC based for potency, impurity, and identification for both drug substance and drug product. Identity, assay, and impurity HPLC. These tests are pretty similar for drug substance and drug products. There are others include moistures by Carl Fisher, residual solvents, by Headspace GC, IC for salt counter ion, and inorganic metal testing by IC or ICP mass spec. This slide summarizes the therapeutic development roadmap in a nutshell using the monoclonal antibody as an example. The figure is courtesy of Brian Kelly of Genentech. First is the identification of a disease-related antigens injected into a host animal to create a fusion hybridomer, followed by cell line development of developing the most optimized cell line, typically a CHO cell, for the production and scaled up of the appropriate monoclonal antibody. Next is a cell culture using a bioreactor or fermentation process to produce the antibody at scale, followed by bioprocessing and purification using a host of different chromatography and affinity chromatography technique. The technical development of CMC processes have some similarity with small molecule drugs such as formulation, QC, and regulatory filing. This slide also come from Brian Kelly of Genentech. The slide overview the bioprocessing and purification processing, starting from the centrifugation to remove cell and debris, collecting the supernatant containing the antibody, and to isolate the antibody using affinity chromatography followed by ion exchange chromatography to isolate the purified monoclonal antibody. The final step is filtration to sterilize and to concentrate the antibody for formulation. Let's examine the quality specification and the mocked batch release results for an antibody product. This table is courtesy of Jennifer Rear et al. From Genentech, coming from a book chapter. The protein concentration is measured by looking at the UV absorption at 280 nanometer. The percentage of purity is often characterized, measured by a host of different chromatography techniques such as size exclusion, ion exchange, and also CE with SDS. Peptide mapping is by reverse phase with UV detection, and also a lot of contaminants from host cells, enterotoxin, and bioburden are measured accordingly. And there's also other attributes such as osmolarity and pH and appearance, which is documented. This slide summarizes the quality attribute of small molecule drug versus biotherapeutics and the QC and characterization tests being used. Note that there are many similarities, though more complex biologics tend to use a multiple technique and additional characterization testing. There are also substantial differences between these attribute and tests. For example, small molecule drugs do not require immunogenicity and structural integrity testing, while it requires tests for solid-state characterization using X-ray powder diffraction or dissolution to measure the release of the API from drug product using UV or HPLC. Here is a summary of the CMC process from small molecule to biotherapeutic. Here we summarize key differences of small molecule and biotherapeutic drugs and explain the CMC process and organization needed to ensure drug quality consistency in new drug development. In the CMC processes, we overview the CMC process in both modality and summarize the quality attribute and testing method. Note that most small molecule development experiences, such as quality regulatory processes and testing methodology, can be translated, though one must always keep track and appreciate the key differences between the two major drug types. Here's a list of recommended references, with the first two from two book chapters. The book on the challenges of CMC regulatory compliance for biopharmaceutical, and also a book chapter by Jennifer Ria and Taylor Zhang in this book over here. QAA. First one, can you clarify the term CMC, which can be referred to processes, organization, or project team? Yes, CMC processes are those developed by chemistry process scaled-up formulation and liquid development and QC, which are required to ensure that the CTM or clinical trial material and final drug product can be produced consistently with acceptable quality. CMC organization in a pharmaceutical company is often called the pharmaceutical science or technical development department, responsible for CMC activity leading to the production of CMTs, CTMs, and the data for regulatory filings. In most pharmaceutical company, drug development projects are driven by multidisciplinary drug project teams and with a CMC sub-teams. Question number 2, there are some profound differences between small molecule drugs and biotherapies. What are the causes of these differences, and what are the similarities in the developmental quality and regulatory perspective? First, small molecule drugs are pure synthetic organic molecule, while biotherapies are complex heterogeneous protein produced mostly by recombinant DNA technology, followed by bioprocessing. The physical, chemical, and pharmacological properties are very different. There are, however, many similarities in the CMC area, such as their quality attribute, such as ID, potency, and impurity profiles, and regulatory pathway. The major differences are in production processes, stability, pharmacokinetics, formulation, and the testing method and specifications. On that note, I'd like to thank you for your attention, and have a nice day.